So while Nick's coming to the microphone, Nick is going to talk about family history risk predictions from the electronic medical record, EMR.
Great. Okay.
Now I can't turn it off.
You got it, Dan? You got it figured out? All right. I'm going to go fast because I kept adding slides. But actually, I just want to take a second and acknowledge that this is the biggest projector I've ever seen in my life. Look at the size of that thing. It's a screen by itself. Okay. This is the size of our room. Look at this thing. This is like the old, what are the ENIACs? Yeah. You'd have a special HVAC just for the projector. I love observation, so what you're going to be seeing is me trying to use observational data in ways that produce real research results. I was inspired because Darwin caught a ride on the HMS Beagle, sailed around the world just to collect observations, knowing that collecting those observations would lead to new discoveries. One of the areas I'm interested in drug discovery, so I always mention McBride and Lentz. Hospitals across the country are using electronic health records. It's making this resource the ultimate source of observational data. And that's the point of the session. George talked about Odyssey. So let's keep going. The question becomes, how do we do observational analysis now that we have trillions of measurements being made every year? Darwin collected a little bit of data. You could see, you can read all of the data he collected, and you can kind of think about it, and he sat down in an armchair and came up with some pretty reasonable hypotheses. But how do we do that now with 1 trillion measurements or a terabyte's worth of data with every sequencing run? We essentially have to train algorithms to find patterns in the data for us, a hypothesis generation phase. Those hypotheses, of course, then have to be validated, just like any good hypothesis is, tested, and then reevaluated. But we're really scaling up the way to make discoveries. So today I'm going to talk about, briefly, a method that we developed to identify familial relationships in the EHR and some uses that we found for that data. EHRs collect a lot of data. I'm going to go quickly because we've been talking about it so much. Most importantly, anyone have any idea, this is a gimme question, which is the most accurate data set of all of these that I put up here? Billing. Yes, of course. Everything else is an afterthought. It's a billing system. And the reason why I'm so excited about these is this table. So I made this table when we were doing this study, and I wanted to use these relationships to study heritability, because I heard heritability was cool and something that people were excited about maybe 15 years ago. And so I went out to the literature, and I found the biggest single heritability study done. There's a lot of meta-analyses. There's lots of data sets that aggregate things together. But for these phenotypes, height, I found one study that, by themselves, they measured 20,000 people. I found another study of diabetes. By themselves, they've measured and recruited 20,000 people, eczema, 10,000 people. And I thought this was great because just in the course of medical practice at Columbia, we have spontaneously collected 300,000 people's heights. Every time you go in there, you get your height measured, your weight measured. You think that's going into the garbage. Nobody looks at it again. But that's not true. I looked at it. And diabetes, we have 140,000 diabetics, and there's only, all of these dwarf the size of the largest single study done of heritability. And this is just one site. Now you add all of our representative institutions together, and we have this enormous data set, really allowing and changing the types of questions that we can ask. No longer is it, is height heritable? We can talk about co-heritability. We can talk about dependencies. We can break these down into different subpopulations that are normally not studied, which we do a little bit here. The problem is these EHR data are not organized by family. We don't ask people who their family members are when they walk into the hospital to treat them clinically. Some people have said we can use some other source of data, people self-assigned race, ethnicity, but in our data set, and probably this is true for a lot of them, and ours is quite diverse, this is over 40% unidentified. But it turns out that there's an extremely reliable source of family relationship data, and it's collected by every hospital, every day, every time a patient's admitted, and that's the Nextofkin data. You go in, you sign a form, you say, who would you like to be contacted in case of emergency? You write down the name, address, phone number. and how you're related to them. That goes into a database, and I went to the database and I pulled out that table, and this is what I found at Columbia. We have 2.5 million next of kin reports or emergency contacts. Number one is mother. 730,000 people reported their mother as the emergency contact. You can see number two is spouse, and a distant third is father. So, and then people trust all sorts of people as emergency contact. You know, their children, their siblings. Sometimes they don't want anyone contacted. Great-grandparents sometimes. And in fact, there's even a couple of examples of a blind date. That's my favorite out of this. I like to imagine they're still together. So we take these data, and there's actually something else we can do with this. This is just because I spent a long time working with an artist on making this illustration. I just like to show it anytime I can. So electronic health records in digital form. Cool. This is how we do the matching. I'm skipping over this part. We match them by their identifying information is the punchline. Okay. So once we match them, we look in our database and we say, how many of these emergency contacts are we actually treating? We find it's about 30% of them are also patients in our database. Great opportunity. Now we have people connected by relationships in our EHR. You can see 386,000 of those 700,000 mother-child relationships. We can take this one step further. And if people come in reporting relationships, we can make additional inferences. If I come in and report my mother as the emergency contact, which I would, she's awesome, and my sister comes in and she also reports my mother, then we can infer that we're siblings. And we can take this out, we can continue to do this until we exhaust all of the relationships we were provided. So we do that, and this is what we end up getting. We get about 500,000 parent-child relationships at Columbia. We get about 420,000 sibling relationships, 200,000 avuncular relationships, a term I learned from doing this study, which technically means of uncle, but geneticists seem to use it just to mean all of those relationships. But whatever, you guys can figure that out. And so we have spouse, cousin, siblings. Look at this. We have great-great-grandparents. We have first cousins once removed. Really fun, interesting relationships that we could use, 3.2 million overall at Columbia. But we repeated this also at Cornell, our kind of sister institution in New York, as part of New York Presbyterian Hospital, and we find another 1.5 million relationships there. We repeat this at Mount Sinai, our competitor in New York, and we find another 2 million relationships there. Lots of interesting dynamic things going on that we could peel into. Overall, we find about 600,000 families across these three data sets. Here's one of those pedigrees. It's been modified. But here's one of those pedigrees pulled out of the Columbia data set. And what we've done is we have, I don't need to tell you guys what pedigrees are, but circles are females, squares are males. They're in a solid outline if we have EHR data on that patient. Dashed outline if we do not. And then I've colored some of them by just some popular diseases so you could see kind of how rich these data sets can be. So that's really cool. Here's another one. This is from Cornell, also changed. But you can see now we have this like marrying of two I don't know technically if they're married, but marrying of two families. And you can imagine the types of studies that we can enable with this data set. We validated this in a few different ways, one of which we had an internal reference set, and I skipped over that slide. But another way we validated it was against genetic data that we had available at a couple of the institutions. This is from Mount Sinai, where we compared the predicted relatedness from our algorithm to their actual genetic relatedness from the available genetic data. And so we are plotting that. So here on the y-axis, we have the predicted EHR relationship, and on the x, their actual shared genetic relationship. And you can see the alpha is a little strong, but it's actually a very strong relationship. You can see up here the correlation between the predicted and the actual. This reminds me that the other word I learned from doing this study is that there is a gender-neutral form of niece and nephew. Does anyone know the word? It's a gender-neutral word for niece or nephew. No one is yet to know this. It's called a nibbling. Yeah. It's the only thing you're going to remember after today, probably. Okay. Anyway, this is really good. There's so much interesting stuff to dive into, but there's too much to talk about. So now that we have these relationships, there's about 7.5 million relationships across these 3 data sets. We want to systematically study heritability. There's a big problem with trying to do that, though. In a traditional study of heritability, you would recruit this individual who has a condition and their family into your study. And you would go to each one of them, and you would apply the same rigorous phenotyping, quote-unquote algorithm. So do they have this phenotype? Do they have this disease? Are they this height? And if you could not measure them, then you would make special note of that, and you could identify them as having this dashed outline. So you either know they have the phenotype, you know they don't have the phenotype, or you know you don't know. In our study, because we're using this spontaneously collected or convenience sample of observational data, we have some confidence that if people are diagnosed or have evidence of a diagnosis, remember diagnosis billing data is the most accurate, that they actually do have that disease or phenotype. But for the ones that don't have evidence of that billing code, we don't necessarily know that they don't have that disease. We're not completely sure. It's probably more evidence than nothing, but we don't know. And then there's people that we just don't have data for. So this introduces an ascertainment bias. Not every individual is equally ascertained in our database, which means that which will bias our estimate of the heritability, if the probability correlates with different covariates. So just to show you that it does, and this is going to be an issue, I did something fun this morning, because I keep saying this, even though I never really show any data for it. But if you live closer to our hospital, you're more likely to get more of your care at our hospital. Makes a lot of sense. And so today I generated this figure to show that that's true. So the hospital, Columbia University, New York Presbyterian, is located in Washington Heights, Inwood, which is this little outlined area. Ignore the heart. That's where I park my car. This little outlined area up here. And so what I did is I took everybody who lives there, and I just counted how many times they've ever been diagnosed with anything. How many diagnoses do we have for them? And put the average over here. So the diamonds are the people who live in Washington Heights and Inwood. And then I just took all of Brooklyn, because it's basically, it's really funny. We take care of about an equal amount of people in this little square as all of Brooklyn, because people who are close to the hospital get more of their care at the hospital. And so here you can see, this is exactly true. We're going to know more about people who live in Washington Heights than in Brooklyn. And so that can introduce a bias when you go to estimate the heritability. The way that we attempt to correct for this bias is by using a very simple kind of naive statistical approach to correct for outliers, where we do repeated subsampling of families. And then we re-estimate and re-estimate until we find nice convergence. And I won't show you the algorithm, but I'll show you the most important result, which is that we introduced bias based on how close people live to the hospital. So we introduced exactly the bias I was just showing you. And as you change that bias factor, this is the estimate using a traditional kind of way, family study to estimate heritability, assuming that your data are correct and don't have this ascertainment bias. The actual heritability was this dashed line at 0.5, and these are the biases that you can see, big swings. Not only is it wrong, it's confident that it's wrong. So that's really bad. But if you do this repeated subsampling approach and let it run to convergence, you sacrifice confidence. You can see the error bars are much bigger, but they remain stable around the actual heritability. So we feel pretty confident that we're getting good heritabilities. We got challenged to test height. Everyone knows that height is 77% heritable because of large meta-analyses like this one. And so after we had fixed the algorithm, we set everything in stone. I closed my eyes, I pressed the enter button, and was very happy when I saw that we estimate height to be 77% heritable in this data set too, and that's true kind of no matter how you cut up the data. These are self-reported race ethnicity groups. Overall, we compared this result to 49 different published studies of heritabilities of traits that we think clearly mapped to things that could be measured by the EHR, mostly just conditions and diseases. And a lot of them we get right. Coronary artery disease, we say 51%. Circulation 2005 article says 49. A major depressive disorder, we said 36, 39% from the literature. Rhinitis, we said 78, 95% in the literature. Some of them we get kind of off. So here's atopic dermatitis. We said 41%. There's this literature study that said 78%. Bipolar disorder, way off from the literature. We say it's 22% heritable, but in the literature, it's up to 70%. So that's interesting. Overall, we find that observational heritability is 20% lower than on average to what's reported in the literature. We're able to estimate using this approach heritabilities for 500 different traits and conditions. You can download all the data essentially at that website. So now that we have this database of relationships, we've been able to estimate heritabilities and validate them. We're kind of asking ourselves, what else can this do? So we've had a lot of clinicians come with some pet projects that have been really cool. So one challenge has been the hospital or gastro and GI docs, I don't know how to say that, GI docs, are curious about the screening rate of celiac disease because they know that you should screen all first-degree relatives that have any evidence of a celiac patient who also show any symptoms. That's the that's the clinical guideline. But they never had a way to evaluate if they were actually ascribing to that guideline. How well were they sticking to that? So we set up this study. Now that we were able to identify these first-degree relationships, and we were able to see that we were actually screening about 50% of the people who should have been screened. So that's cool. And there's some extra in-depth stuff that's kind of fun, and some predictors that we found of whether or not you get screened, if you see a GI doc. And so that was cool. Another study we did in collaboration with some cancer researchers was they were doing this study where they're screening people to go breast cancer testing. And they were using a very simple approach to look in the EHR. But family history data is not always available in the EHR or well documented. So as a replacement, we substituted our algorithm to identify first-degree relatives with any evidence of breast or ovarian cancer, and basically synthetically computed this score. And we found some additional patients' sensitivity was actually pretty low. That's to be expected because not everyone has this relationship data available. But it kind of exemplifies that this is another potential use of these family history data. All kinds of interesting ethical questions come up, but we can talk about that in the discussion section. And then the final thing is that using the EHR allows us to design heritabilities in interesting ways. This caught me by surprise because now it's counting up. And I was like, wow, that's weird. But I get it. I'm getting the cane. But anyway, so we did this large-scale, in-depth analysis of stroke. And
this is your 1 minute morning.
oh, thank you. Thank you.
Your minus 1 minute morning.
Oh, okay.
Just keep going.
I'll talk in reverse.
Talk fast.
We set up, so basically, we want to do machine learning to identify stroke patients in the EHR. So what we did, because it's hard to determine what data set to use, it's highly dependent on the data, highly dependent on the model. So we just decided to use all possible data sets that we could think of, all possible models, and compare them all systematically. We did that. I'm not even going to go into details. They perform very well, some of them. And probably the most important thing is that you can train these algorithms to perform very well, even with very little data. So even with only 70 to 350 stroke patients, we can predict stroke pretty accurately across the entire database. And like I said, I'm not going to go in details, but we validated it in the UK Biobank data. And this is that positive predictive value or improvement over expected in the top 50 patients that are ranked that are predicted to have stroke in the UK Biobank data, and how enriched they are for patients who really do have stroke. And I should say that this actually doesn't use any diagnoses codes for stroke at all. So it's actually inferring it from all other places. No direct evidence are used in these algorithms. And then finally, because we are able to compute a risk factor or this probability of stroke for every patient in our family histories or our family relationships, we can estimate heritability of stroke, which we couldn't do directly with the stroke data that's available in our EHR, and it produced accurate results. With that, I'd like to thank the first authors and everybody else. Thank you.
We're right now at the point where we should start our panel discussion. So if there are specific questions for Nick about, you know what, just we'll start the panel.
No
so
questions.
Go back to sit, and if people have questions for you, they can ask in the panel. So this whole session from Josh, Mark from this morning, and then George and Nick from this afternoon, open for questions, discussion.